The antibody molecule can readily be cleaved into functionally distinct fragments. The protein domains associate to form larger globular domains. Thus, when fully folded and assembled, an antibody molecule comprises 3 equal-sized globular portions joined by a flexible stretch of polypeptide chain known as the hinge region. Each arm of this Y-shaped structure is formed by the association of a light chain with the amino terminal half of a heavy chain, whereas the trunk of the Y is formed by the pairing of the carboxy terminal halves of the 2 heavy chains. The association of the heavy and light chains is such that the variable heavy and variable light domains are paired, as are the constant heavy 1 and the constant light domains. The constant heavy 3 domains pair with each other, but the constant heavy 2 domains do not interact. Carbohydrate side chains attached to the constant heavy 2 domains lie between the 2 heavy chains. The 2 antigen binding sites are formed by the paired variable heavy and variable light domains at the ends of the 2 arms of the Y. Proteolytic enzymes, proteases, that cleave polypeptide sequences have been used to dissect the structure of antibody molecules and to determine which parts of the molecule are responsible for its various functions. Limited digestion with protease papain cleaves antibody molecules into 3 fragments. 2 fragments are identical and contain the antigen binding activity. These are termed the Fab fragments for fragment antigen binding. The Fab fragments correspond to the 2 identical arms of the antibody molecule, which contain the complete light chains paired with the variable heavy and the constant heavy 1 domains, but was originally observed to crystallize readily, and for this reason was named the FC fragment, for fragment crystallizable. This fragment corresponds to the paired constant heavy 2 and constant heavy 3 domains and is part of the antibody molecule that interacts with effector molecules in cells. The functional differences between heavy chain isotypes lie mainly in the FC fragment. And that's the antibody molecule can readily be cleaved into functionally distinct fragments. Have a great day.